Please be seated.
Hereby, I open this academic ceremony in which Giuseppe Marrazzo will defend the academic thesis Corpore, Cognoscere, Computational Insights into Human Body Perception. May I invite you to present a summary of your study and the conclusion of your thesis.
Dear Prorector, esteemed committee members, supervisors, family, and friends, thank you all for being here today. I am excited to share the results of the work I've done in the past years, which I've titled Corpore Cognoscere, a Latin phrase meaning to know through the body. Now, let's take a moment to look at the image on the screen. You immediately see the shadow of two people holding hands. If you look closely, you can infer their biological sex. The left figure appears male, the right female. And their raised hands probably signify that these two people were happy in the moment this picture was taken. Finally, if you know me well enough, you probably have recognized that the person on the left is actually me. Now, this is an interesting phenomenon. The brain is able to infer the biological sex, the emotion, and the identity of bodies, of human bodies, just from their silhouettes. But what is in these shapes that powers these inferences? Is it the contours and edges? Is it the relative position of joints and limbs? Or is it, for instance, the realism or the plausibility of the postures? Today we will unravel the key features that our visual system exploits to basically know through the body. Therefore, this thesis aims to deepen our understanding of how human brain processes visual information coming from human bodies, and it does so in three main empirical chapters. Specifically, in Chapter 2, I will show that body perception is not a static process, but can be shaped by contextual demands. Chapter 3 will focus on unraveling the characteristics or features, if you want, of the human body that can significantly explain patterns of activation in the brain. And building on the results of Chapter 3, Chapter 4 will address the question of whether the plausibility of the postures or the movement is actually a factor when describing the representation that our brain makes of human bodies. So without any further ado, let's look into the results of Chapter 2. So traditionally, models of object perception have often conceptualized the brain responses to different types of objects as categorical. Or if you want, these models suggest that specific brain regions are deputed to process one specific type of stimulus. Like, for instance, the extrastriate body area is, according to this model, rigidly dedicated to process human bodies. And it does so in a way that is relatively stable and therefore independent from external factors, like, for instance, contextual demands. So to check whether body representation is solely categorical and therefore independent from, for instance, task demands, in Chapter 2 we showed the same picture to participants and we asked them to judge. You asked them two different questions. In one case, we asked them to judge the emotion depicted by the posture, which could have been anger or happiness. And in another case, we asked them to simply name the shape that was superimposed onto the body, which could have been a circle or a square. And we did this while we recorded that brain activity using functional magnetic resonance imaging. We analyzed the data by basically instructing an algorithm to classify brain responses based on the type of task that the participants was performing. And ultimately, we found that the brain, by simply looking at the brain activity, we can understand the type of task that the participants was performing. Or if you want, we can decode the task performed in several key areas like the inferior parietal lobule, the ventrolateral prefrontal cortex, and the extrastriate body area, signifying that also the so-called category areas like the extrastriate body area can flex according to what is asked to do and not necessarily only to what is shown. So what does this mean? This means that by showing exactly the same images, we can basically, by changing the type of question, find very different brain patterns. We can find these differences in key areas like the extrastriate body area, which is a sensory areas, and the ventrolateral prefrontal cortex, which is seen as a high-level control area involved, for instance, in deciding which type of information the brain should focus on. And the fact that these two areas show very similar representation could indicate that sensory and control areas work in tandem in a way that basically they talk to each other. So basically the sensory cortex provides the raw input and the high-level control areas can stream this data to highlight basically what the brain needs to do, or if you want, highlight the features that the brain needs to perform the task at hand. So this tells us that basically the task context does matter, but it does leave open the questions of whether which type of specific visual cues is the brain tuning into when it's adapting these representations. And to address this question, in chapter 3, we used computational modeling combined with the ultra-high field functional magnetic resonance imaging to understand which characteristics or features, if you want, of the human body drive responses in body selective regions. In this study, basically, we generated many different poses using an artificial neural network, and from each of these poses, we extracted the 3 different types of information, or what we called features of the stimuli. Let's explain a little bit what these features are. A Gabor model is a model that basically mimics early visual cortex processing by detecting local orientation and spatial frequencies. Now, since it operates at the level of the pixel value, so before any notion of body shape or body parts, so on and so forth, this is what we call a low-level model. A key point to the model is a model that for each image extracts the XY coordinates of position of shoulders, elbows, hips, knees, and so on and so forth. Now, while this representation captures recognizable body parts, like you can see arms and torso and so on and so forth, it does still depend on the viewpoint of the camera, so it does treat the human body as a bidimensional object in this case, so the image projection. Lastly, the key point 3D model instead treats the flat image as an instance of a 3-dimensional pose. So this abstraction is independent of the viewpoint of the camera, making it conceptually the most advanced representation. Finally, what we did was using a mathematical model to ask which type of features can, or combination of these features can best predict responses to stimuli, to this type of stimuli. Now, to explain a little bit how we did that, I will try to spend a few more words about this. Well, starting from this representation, those 3 models, what we did was basically generating synthetic brain response pattern to these stimuli. Now, by comparing these synthetic responses to these stimuli to the real responses that the brain produces when we show these pictures to the participants in the scanner, we can basically compare them. And if we basically find that these images are similar, these responses are similar enough, we can basically conclude with a reasonable degree of caution that the type of the computation that the brain is performing is similar to the type of computation that our computational model are performing. So let's see what we find here. So we find that a combination of these 3 models can significantly explain responses in ventral visual cortex. Specifically, if we assign to the relative importance of each model a color, we can basically create a map that divides the relative importance of each model across the cortex. And this is basically what this map is depicting. The first thing that we see that there isn't a sharp division of the information across the cortex, but rather this information is represented following a gradient of colors. And if we zoom in the back of the brain, where early visual cortical areas are located, we can see that there is a blue dark magenta patch dominating, signifying that the low-level representation or the Gabor model is leading this representation. If we zoom instead in the EBA, we can spot a subtle mix of color, which confirms that EBA itself is not one feature or another, but rather a combination, which in this case is favoring the 2D joint structure. What does this mean in practice? It means that the brain is not segregating low-level processing from mid-level or high-level processing, but in isolated patches, but it's working along a smooth continuum. And where in the case of the EBA, we see that the EBA is favoring 2D joints representation. And in the posterior part of the EBA, this representation remains tied to the image projection. That is, it does change when you rotate the viewpoint of the camera, when you rotate the camera. But as you move more anteriorly, this representation becomes more viewpoint invariant. Finally, I would like to focus the attention of what I did in chapter 4. Now, in chapter 3, I just showed that the joints position is one of the drivers of the responses in body selective regions, and specifically in the EBA. So what this means is that the relative arrangement of the joints seems to play a role when it comes to process body stimuli. So we asked, what happens if we break this arrangement? So to answer this question, we designed an fMRI experiment in which we showed participants videos of avatars where the normal biomechanical constraints were violated. And just like in chapter 3, we tested 3 computational hypotheses. We started from the key point 3D model that we introduced previously. And from this representation, we built a model which is capable of measuring in a quantitative way how possible poses are different from impossible ones, starting from their postural configuration. We call this model the SIM-DIST model. Lastly, we also included a categorical model, which is very simple in its definition because it just has 2 features, basically. But it's very high level when it comes to the implication for brain representation, as it assumes a clear separation between the possible and impossible category based on semantic labels. Now, we used the same mathematical framework that we introduced in chapter 3, and what do we find? Again, we find that there isn't a clear separation among features, and that the EBA is sensitive to biomechanical differences to the stimuli, as the blue and green patches here in the bottom row show. Once again, rather than acting as rigid body modules, EBA is representing a continuum of features, integrating in this case our sense of what bodies should look like and also how they are supposed to be moving, so whether or not they obey to the laws of human biomechanics. To conclude, across chapters, we have built a cohesive picture which points towards a dynamic and integrative model of body perception, where the occipital temporal cortex and specifically the EBA is dynamically combining multiple types of features with different level of complexity. Low-level features, mid-level features, and high-level features are all very well represented in a way that is integrated hierarchically. And in practice, the fact that already relatively early in the visual stream a multitude of features are represented, and that this representation seems to be task-dependent or anyway can be affected by external factor, means that our brain is not simply passively seeing bodies, but it's actively interpreting them. And it's basically doing so by adjusting its computation to serve specific behavioral goals, like, for instance, reading intentions, emotions, or understanding that the person on the left here is me. I thank you all for your attention, and I give back the word to the prorector.
Thank you for your nice presentation. The opposition will now be opened by Professor Pavlova, who holds a chair in social neuroscience and is associated with the Department of Psychiatry and Psychotherapy and the Tübingen Center for Mental Health, and holds the chair at Eberhard Karls University of Tübingen. The floor is yours.
Thank you so much. Thank you, Giuseppe, for your presentation. I do have one general question first. Could you formulate in 1-2 sentences the novelty of your work? For example, in chapter 1, you are saying that brain activation is pretty good modulated by task demands. But this is not new. We know this already from many other domains, not only from body, shape, and emotion perception. Thank you.
Highly esteemed opponent, thank you very much for your compliment and questions. And yes, it is actually not new to find like this type of modulation. The thing is that it wasn't found specifically for bodies, namely in the case of and especially bodies emotion expressing affective information. So the modulation was found previously in a high-level visual cortex when it comes to the in prefrontal cortex, but not in high-level visual cortex when it comes to modulation driven by task itself. That's what we meant in chapter 1. Yeah.
I'm really not pretty sure. This is about chapter 1. I'm not pretty sure this is like this, but could you formulate in 1-2 sentences the novelty of the whole work? Because you have several chapters, right?
Yes.
And they are not really strongly connected with each other.
Okay.
So my problem when I also wrote my report about your work was that it was a little bit mosaic. So what I want now to know, what is just the main message you are sending to all of us?
Yes.
Merci.
So if I should summarize in like a couple of sentences, I would say that the novelty of the work comes from the fact that there is a shift from categorical representation, if you want, which is still a huge part of brain processing, obviously, but rather than focusing on categories, which in our view is a step further in the processing. So the brain is basically forming categorical differences starting from specific features of the objects. So in this case, our interest was to show that the brain is not only functioning by separating objects in categories per se, but these differences are arising from specific features of these stimuli. For instance, in chapter 3, I show that by looking at images, the relative distance of the joints is one of the main drivers, let's say, but by looking also at either type of processing, for instance, when body starts moving and stuff, also differences in biomechanics can trigger like differences in body selective regions. So the neurons in EDBA probably are tuned to specific characteristics of the human body, and then the fact that when we contrast these across different domains, object domain obviously, we manage to find differences probably stem from the fact that the features of the human body are different from other type of features from other objects. That is like the...
Okay, when you were doing fMRI analysis, how did you define the regions of interest?
Yeah.
Did you conduct whole brain analysis, or you are just were looking for specific areas like ABA and others, which are many not only this, what you are presenting here.
Yeah.
So what were your criteria?
Yeah. So the analysis was performed in two ways, basically. So we had an independent localizer, which we defined based on category model, right? So the fact that different areas respond more to specific type of stimuli compared to another. But when it comes to processing, to selecting the voxels to perform the analysis, we had we took the main effect of the localizer. We didn't only select responses from EBA, but was rather distributed across the ventral visual cortex. So that was in chapter 2 and chapter 3 at least what we focused on.
I'm very sorry because this is my first time in the Netherlands. I am not familiar with the whole procedure, so I'm unsure, so to say. I'm very sorry for this.
You have time for one last question.
Okay, thank you very much for telling me. But you didn't reply to my previous question. So I mean, there are several areas which are specific for body motion and emotion from body motion perception, right?
Yes.
And so why you just limited yourself to these specific areas? And how did you take into account that this activation, for example, in the EBA, is not modulated by other areas for brain communication, for brain networking? I mean, there are many connections in the brain, and now our modern neuroimaging is specifically focused on brain connections, not only on activation in one or two specific areas. Merci.
Yeah. Thank you very much for your question. So we had like a localizer, right? So we used that localizer to select the responses that from ventral visual cortex. And we focused on ventral visual cortex because we wanted to kind of understand which type of features are driving the responses. So which type of feature that you can extract from the stimuli in a bottom-up fashion can drive responses in body selective regions, which for the type of stimuli that we used, the strongest response, or if you want, the best localization happened to be in EBA. And therefore the work then focused on the EBA. We also found performed analysis on the FBA as well in the chapter 3. In chapter 4 we did not manage to localize FBA. And when it comes to the motion component, for the type of analysis that we wanted to perform, the motion energy between the two categories was exactly the same. So when it comes to the motion processes per se, the differences that were arising from the stimuli could not have come out from motion itself. So because when it comes to the videos, the two stimuli, the motion energy between the two different categories, which is the focus of the analysis basically in chapter 4, was the motion energy was the same across the two categories. So we built the stimuli controlling for the motion energy.
This time I will have to tell you that the time is up. Thank you for your questions, Professor Pablo. And I would like to give the word now to Professor Jansma, who holds the chair in cognitive neuroscience of language and of language processing, and who is at our Department of Cognitive Neuroscience, our faculty in our university.
Thank you, Prorector. Dear candidate, first of all, let me congratulate you to your great thesis, also to your supervisors. I read it with pleasure, and I think I learned a lot, also new stuff in terms of contextual impacts of task and demands, and also especially also on encoding models and combining different ones and finding hierarchies. So I really like your work. It's also published well already. I also heard that recently you even got chapter 4 out, or at least in revision, and that's nice because my question also relates to this chapter 4. And it's a bit about the concept of stimuli and also the methods you use. So usually in vision, like old stuff was presenting static pictures. You are more advanced going towards video clips. But there, and you are interested in, of course, feature selection, but also towards the ultimate end game is us interpreting other people's motions, right? So I just wonder, and that's a conceptual question, when you present video clips that takes 2 to 3 seconds on average, if I remember correctly, the event unfolds over time. And that reminded me of language, where actually when I hear a sentence, word by word come in, and I do some guessing of what the sentence will be about. I predict eventually the ending, and at one point I might be right in my prediction or wrong, like the pizza is too hot to drink, right? And you presented these video clips of possible, impossible motions, actions, and I wonder whether the point of interpreting this as being impossible or not is right from the beginning or unfolds over time and is there at a certain point in which might be divergent across trials.
Highly esteemed opponent, thank you very much for your compliments and questions. This is a very interesting and important questions actually. And we, by looking at, so I try to kind of understand whether this was happening somehow. I did not have the time resolution to understand this processing itself. We did try to look into layer activations, like to see whether these like differences was more input driven or was like more feedback from higher areas. But we did not find statistical significance across layer analysis. We did find a trend in which we seems to look at when we look at the SIMDist model, which is a model that kind of measures distances, like across how distant are from each other possible versus impossible stimuli, which is constructed from the postural configuration. In that case, we did find some sort of a trend between like this model and the categorical model in inner and superficial layers. So where there was like kind of a switch of how much variance was explained by the two models. So more from the SIMDist in the inner layers and more for the categorical ones. This in the EBA, right? EBA.
Yeah, so just to interrupt, that's completely right. But I wonder, when you have the video clips, would it be possible to actually pin down some events where the interpretation is unique versus ambiguous, so that you basically see some sort of conceptual shifts in interpreting the video clips as being impossible or possible? Would you see a way to do this in fMRI, or do you need other methods for doing that?
I mean, if I would want to pin it with fMRI, I would probably extract the most informative frame probably and try to see, because in fMRI it's very difficult to obtain that level of temporal precision, but we are actually running in the lab a couple of pilots with these same videos, and we did see as early as 170 milliseconds already a difference between the possible and impossible stimuli.
I see. Okay.
So it is some, but it's work in progress. We have very few results about that, and we are also kind of trying to design a study to investigate causality with this same stimuli using TMS as well. But I can say that when it comes to this specific stimulus set, because of the bending of the joints, it's pretty clear already from frame number 1.
Okay, that brings me to another point. So I once saw the clips, I think, if I remember correctly, and my first interpretation was for the impossible ones, ooh, there's someone hurt, there's a broken leg and like twisted elbows. And actually then it's not just impossible, but it's much more what I see and what my brain interprets there. Probably related to the colleague's question. So if you would have the opportunity to look also in other brain areas, I understand that your field of view was limited, but where would you look at to see what else might be involved in interpreting these actions as being impossible?
Yeah, I mean, for my next study, actually, I am trying to look into inferior parietal lobule, which is involved in the mirror neuron like system, the ventral, the premotor cortex as well. I would expect to see some, like they say, the research question of this other study, it's basically try to understand what comes first, action or biomechanics. Namely, do you need biomechanics to understand action, or do you need action to understand biomechanics, basically.
Yeah,
so,
I see. And probably my last one, so I have several, but probably limited. So you repeated the stimuli, I think, three times per run. You had 2 sessions, or the repetition actually is 6 times.
Yeah.
And in the old days, we had something like an adaptation paradigm in fMRI analysis. Would you consider this worthwhile to look into that, whether adaptation to highly frequent, probably well-learned, usual known action is different than adaptation to something that is new and not learned yet?
It would be actually a very interesting thing to look at. I did try to see whether there was a difference between the sessions, and I didn't find any difference within the voxel that we selected for the analysis. But I do think that this is a very interesting thing to look into it, and probably by using stimuli where an internal model needs to be updated, probably the adaptation effect would be different compared to stimuli that are possible. I mean, this is my guess at least.
Yeah. Okay, thanks a lot. I give the word back to the prorector.
Thank you, Professor Jansma. The opposition will now be continued by Professor Goebel, who holds a chair in cognitive neuroscience at the Department of Cognitive Neuroscience, FPN, our university. The floor is yours.
Thank you, Prorector. And dear candidate, I also want to first congratulate you for this great thesis. It was a pleasure to read. I learned, like Professor Jansma, a lot. And I was also not only fascinated by how you tackled the content, but also about your methodological expertise to do that. For example, using encoding models at a state-of-the-art way, it was a pleasure to see how you compared models using bandit-rich regression. A not so standard tool was really great because you had a solid way to find the unique contributions of each model. That was really nice to see. And of course, there are always questions. Mine are more driven by curiosity, I think like the questions before. Especially, I'm working on an area which is close to EBA, MT, Human Motion Complex. And I always wonder when I see work from your supervisor's group, which I want to congratulate actually for that work. Oh, this is MT, but then I see, no, it's not. It's maybe neighboring, it's overlapping, but it must be related somehow, right? And of course, if you look not just at static bodies, you have motion, right? And then I wonder whether we could, whether you know how from connectivity, from inputs, from whether, for example, MT feeds into EBA, or whether they both get input from early visual areas, whether you know something about how these two areas relate. Is one more first stage feeding in the next, or are they more independent? Or what is known about that, and what do you know about that?
Yeah, this is a highly esteemed opponent. Thank you very much for your compliment and questions. Yeah, the relationship between EBA and MT is, I mean, these two areas are connected, definitely, in the sense that it has been shown also from studies on primates and stuff that there are like multiple areas. It has also been found for motion and bodies in macaque, as Professor Vogels probably can tell us. It's the relationship between what I would personally focus on in the chapter 4, which is the only chapter that had motion. As I said before, it is kind of difficult for me to disentangle the MT, because, as I said, the motion energy between the two categories of stimuli were different. And also the research question wanted to kind of include the motion as a means, as prediction, right, for action, and see basically how that would kind of relate in the process within the EBA. So I would, if I have to give an interpretation to this, I would depute the differences to EBA in this case rather than human MT.
Mm-hmm. Thank you very much. I think what you did in your thesis is remarkable, because you tried to track low-level, mid-level categorical features to understand better how these all interact. And you present a nice model at the end. One way which helps to understand the feature space, the earlier mid-level and categorical too, which we did in MT, is to map the organization of this area, like by using simple stimuli, like moving to the left, right, up, down, and so on, and map it out at a laminar and columnar level, right? And I wondered, because you see these beautiful gradients where you see more the lower level features, more posterior, and then more anterior, you see the more higher level ones, whether it would be a good idea to try something similar, to use some stimuli. You could use body parts moving or some global movement, configurable movement, to try to find not only in the laminar level, which you studied in chapter 4, but also at the tangential cortical level, to get a bit closer to what these features really are, which you try to find. And maybe my approach is naive, but it would be maybe possible to come from simple features like body parts and motion direction to more complex ones and map them, and then find out whether you get organization maps and see how they are organized and what is represented in this area. Would you think this is too naive, or would you think this is useful to do?
No, I mean, I think this is very, very useful to do, and thank you very much. I will be taking notes about this. Yeah, I mean, I do think that see what is the interplay between the processing of the human body parts or bodies in general and motion. I mean, we call it the human MT for a reason, right? So it is kind of a very interesting thing. I do believe that. I mean, there is some work. I mean, I think you were referring to the work from Alessandra, right?
Yes.
Yeah, which is actually very, very interesting. I participated also in a couple of in one of her experiments with the motion, the switching of the direction and stuff. That was very cool. But yeah, I do think that. I think that we can, about this, learn a lot by reading the work of Alessandra rather than necessarily like designing a new experiment. But by doing this, I think this is, yeah, I mean, we will definitely think about.
Thank you very much. And talking to Alessandra would be good for that.
Yes.
I want to come to chapter 4, where you used laminar fMRI, which I was also very happy to see. The results were not significant. There were some trends. Yeah. But I wondered about your hypothesis, which you investigated. Namely, before you looked at the results, what the left and the right hemisphere are doing, you very specifically argued that the upper layers will be more for the categorical ones, if I remember correctly, and the deep layers more for the featural and the biomechanical plausibility aspects. And I wondered, how the hell did he came up with this hypothesis? Yeah. Normally people assume, at least in small tangential parts of the cortex, say columnar regions, that you have a very similar response, and other areas top-down might influence that, but that this kind of small patch might correspond to one of the features which are coded in these areas. Yeah. It's very simplistic, I know, but let's follow this logic. And then you have this idea that the layers have a very distinct features, right? They would not be like tangentially sorted, but your features are kind of in depth sorted, which I found very surprising. I want to know, do you have some evidence for that or some ideas where this comes from?
No, I mean, I was simply looking at the literature about layers in general. Mm-hmm. So the fact that middle layers are more input-driven and deeper and superficial are probably receiving input from higher order areas. So I had an hypothesis of what I believed was a low-level model, a middle-level model, and a high-level model, and therefore I have distributed like the importance across the different layers. So this was basically the rationale behind. Yeah.
what speaks to your point is that we see more and more in higher areas that the connectivity in the layers is of course different, and that means they get influenced by different input. So it could well be that if you look at your gradient EBA, that you really find that other areas feed into the upper layers and change the responses and so on. So I found it quite a very nice idea to pursue.
It would have been nicer to find it statistical significance.
Yeah, that's true. That's true. But at least you had some trends. Do you have still time, Prorector?
Very short.
Okay. A very short question to chapter 2. Unfortunately, it didn't find the emotional categorization of the anger and happiness. And I wondered why that, and there you looked also in prefrontal areas, ventral medial prefrontal cortex.
Yes.
And I was a bit surprised that you did not find any significant differences there. Do you have a reason why that is?
I think probably the different cognitive demands across the two tasks, because the task effect is known to like engage, like the attentional load is more and stuff. So the differences across the different emotions, also in ventral prefrontal cortex, in the explicit condition, were very much flattened out, right? So this is probably the effect of the task itself. And I was also
overwhelming smaller effect.
Yes, and I was also very surprised not to find that even at low level we did not find differences, because the postures do look different. For anger, like the arms are close to the body, whereas they point up for the happy condition. So even in that case, we did not find differences, which I found puzzling. Yeah.
Me too. Thanks so much for your answers, and I give the word back to the prorector.
Thank you for your questions. The opposition will now be continued by Professor Vogels, who holds a chair in neurophysiology at the Laboratory for Neuro and Psychophysiology at the Department of Neuroscience at the Catholic University of Leuven. The floor is yours.
Thank you. Dear candidate, it was a pleasure reading this thesis. I knew some of the work, of course, because we have a common project. So, but still, it was very nice to see all the details, and also just like the previous, like Professor Goebel was saying, I like a lot the analysis you did. So it's really state-of-the-art analysis and methodology. But of course, it's my task now to ask some questions. I have, well, a long set of questions, but of course we're limited in time, so I'll do not a random, but some sampling of the questions. So I will start with your chapter 2, so the task experiment in which you manipulate tasks. So you have an emotion categorization task versus a shape discrimination task. And you find modulations by task in several regions of the brain, including EBA. Now, of course, it's always interesting to know what is causing these task differences. Because if you look at your tasks, your two tasks is not just emotion versus shape, but there are also other differences. For instance, in the emotion categorization task, you present a body, and most of these emotions, if you want to categorize these emotions, you have basically to rely on the posture, on position of the limbs and things like that, which are, well, in the image, external, let's say, external features compared to the shape where you have a central stimulus. So there's a difference in spatial attention probably by the subjects. And then another factor, potential factor, which might be good to discuss, I think, is the difficulty. So you do a nice behavioral analysis of your behavioral data, and you show that the emotion categorization was more difficult. Longer reaction times, lower proportion of correct responses. So there's also a difference in difficulty, and that also can impact activations, as we know. So to what extent can these two factors, spatial attention and difficulty, explain the modulations that you see? And maybe there are also other factors, because it's...
Hi, esteemed opponent. Thank you very much for your compliments and questions. So, yeah, I have been thinking about this for a lot, to be honest, because I do think it all stems to what is the definition that we want to give of task. Namely, so I mean, task is just putting, like in the case of emotion, like a semantic label, define this as anger and happiness, and shape a circle or the square. So I could not like give these. So the participants were instructed to fixate onto the shape, which was on for both tasks in both for at all time, and therefore like the differences cannot be like driven by differences in visual input in that case. Yeah. So, but when it comes to the fact that the emotional task is more complicated, albeit also for the emotion task, I think the accuracies were around 90% or something. So it was high, were like high accuracies. I do think that the attention does play a role in this anyway. So task difficulties can probably, because the brain needs to engage more cognitive resources, kind of also show the pattern of the representational dissimilarity matrices in explicit task, in which the attribute differences were completely flattened out because probably the cognitive load was higher in the explicit task compared to the implicit one. So it does, in my opinion, play a role. And with the attention itself, attention is required in this case to perform the two tasks, right? So it is probably one of the drivers as well in the response, in like defining the differences between the two tasks. Yes.
Thank you. A related question, or somehow a related question, of the same experiment, has to do with your RSA analysis that you did.
RSA.
Yeah. And where you find an interesting pattern of similarities for the emotion task in VLPFC and in EBA.
Yeah.
And as I understood it, the different emotion and skin color categories, they voted pretty similar activations.
Yeah.
In the emotion task.
In the explicit
task, they're
yes.
more dissimilar in the shape task.
In the shape task.
I would expect the opposite, because in a motion task, basically, you need to, discriminate or categorize the emotions. So then the different emotions become important, like happy versus angry becomes important. So you would expect to find dissimilarities in the response, and not more similarities compared to the shape categorization test. Do you have an explanation for that finding?
Yeah, it is an interesting question, obviously. I do believe that this is what was defined as the task effect itself. The fact that naming the emotion, the fact that we already labeled, so we gave them a fourth choice between the two category, kind of made the task, it wasn't required to kind of make this level of high-level categorization, and therefore the fact that they were engaged in performing the task has flattened out the responses, like stimulus attribute specific responses in the explicit task. But while they were focusing their attention on something different, namely the shape, while supposedly ignoring the posture behind, then you start saying that the emotional processing, or like differences across start to arising, pointing towards the fact that maybe this is more of an automatic processing rather than something that you need specifically to focus on. Yeah.
Well, the reason I find it a little bit peculiar, what you found, is that if you look at other experiments, many experiments on task modulations have been done in the past.
Yeah.
And for instance, if you ask subjects, if you present subject with identical stimuli and they have to judge, for instance, the speed, or they have to judge the shape, what you see basically is that when you're judging the shape, you see more dissimilarities for the shape compared to the speed, and vice versa when you have to judge the speed. And you seem to find the opposite, so that's why I've found it so strange.
It is, the thing is that there was, the task, as I said, wasn't so difficult, because the accuracy was 90%.
Also in those experiments, it's not that difficult, so.
Yeah, but in this case, probably they were, especially for the position of the arms and stuff, could also be that they were categorizing the position of the arms directly. So you just see that, and you while ignoring the emotion, associating like a specific label to the position of the arms, or to judging the posture rather than judging like the emotion itself. And because like the picture were all pretty similar to each other, there was some variability. They were pretty similar to each other. It could be that in the end you did find this pattern, let's say.
Okay, thank you. I have to stop.
Thank you, Professor Vogels. I'm sure that there will be more time to discuss at a later point in time. But now I will give the opposition to Associate Professor Giancarlo Valente, who is at the Department of Cognitive Neuroscience, has his expertise in statistical and computational modeling of neuroimaging data. The word is yours.
Thank you, Prorector. So, dear candidate, first of all, congratulations. Congratulations on this really nice thesis, and I would like to extend the congratulations to your supervisory team. It was a really nice thesis to read, and I have a good emotional response to well-conducted analysis, and I really appreciate a lot the fact that you are combining interesting content questions with advanced methods. So you're not limiting yourself just to standard tools, but you're really going at the state of the art, pushing the boundaries. And you start with decoding, and then you go also to encoding models with this banded ridge regression in which you compare multiple models. Now, as you may expect, I have a question on the methods. So since we're talking in Latin, so this will be like the dulcis in fundo for this opposition. Hopefully it's not the in cauda veneno. And this is about, as you may have guessed, about model comparison, right? So model comparison is at the heart of what we are doing. We are testing different models, and we see which one is more supported by the data. And I have for you just a very open question, so there's no right or wrong. It's more I would like to think together with you. And model comparison has been done a lot in linear regression literature for almost a century and a half. And when you do things in samples, you get a sample and you try to see whether your predictor explains the samples. There's lots of caveats that have been given that the coefficient of determination, how well you explain the data, is inflated when you have lots of explanatory variables. In your models, you have lots of explanatory variables. This is the beauty of it. So you don't limit yourself to one or two factors, but you get like hundreds for the KP2 3D and KP 3D, and you have thousands for the Gabor patches. Yeah. And what is nice is that then you transform this from explanation to prediction. So you split the data, you test on the data set, and you train on a data set and test on the other. So you kind of leave some data out, and ideally this should circumvent the problem of having different dimensions. What I would like to probe a little bit with you is that actually it has been shown also in the statistical literature that whereas there is an upward bias in the coefficient of determination when you have more explanatory variables in your training data, so the more things you throw, of course the data are going to fit better. There is actually a downward bias in the coefficient of determination of out-of-sample data. So the more complex your model is, the more likely it is that it overfits the data, and therefore the performance will drop down. So I would like to play a little bit the devil's advocate. And let's say by reading the results, for instance, in chapter 3, and also chapter 4, one observes that, for instance, KP2D always wins almost everywhere over KP3D. And you argue that this is because maybe the features that are in KP3D, which are not in KP2D, are redundant and are irrelevant. It also, KP2D always wins onto versus a Gabor patches model, like purely visual model when you get lots of features. When I'm asking, couldn't this just be due maybe to the fact that the model that wins most of the times is the simplest, is the one with the fewest feature? And maybe, I would like to hear your opinion on this, and then if you acknowledge that there is a problem, which ways would you think of, how would you address this problem?
Highly esteemed opponent, thank you very much for your compliments and questions. Yeah, this is a very interesting question, and yes, so just to repeat, your questions is about dimensionality issues when it comes to partition the variance, right? So, yes. One could argue that the principle of Occam's razor should kind of favor it, right? The choosing the simplest model when the explanatory power is similar between the more simple model and more complex model. The answer to this question is there is a short answer and a long answer. I'm going to give you the short answer, and then I'm also going to give you the long answer. Okay. So the short answer is that there are ways to correct for this. When we published the first chapter 3, we did not know already about these ways to correct for this, but there is literature about how to adjust the R-square for the out of sample when it comes to partition the variance and take into account dimensionality differences across like the models, even though the bounded ridge claims to, like, by regularizing differently, independently, the different feature spaces, takes into account like these differences, collinearity, overfitting, and so on and so forth within the features. And because bounded ridge uses as a metric to kind of obtain a coefficient of determination, they use the product measure. The product measure is also affected by this bias, especially in the out of sample. So I would personally rerun the analysis and let you know if the things change by recomputing the R-square, but now comes the long answer, sorry. The difference is that we, while methodologically speaking, these things are completely true. Obviously, there is the statistics, and the statistics obviously give us a lot of insights in what should be done and how it should be done. We still need, the statistic does not know that we are performing computational modeling on fMRI responses in response to body stimuli and so on and so forth. But because research has shown us that, for instance, early visual cortical areas respond to, like, the PRF are tuned in a way that they respond to like spatial frequencies, orientation, and so on and so forth. Seeing that in chapter 3 the Gabor model still fits a little bit better than the others, by looking at the gradient we can see that there are blue patches and dark magenta patches in early visual cortical areas, points towards the fact that even though there is this bias, and it's pessimistic, right, in the case of the out of sample, towards the most complex model, the most complex model in that case still is, it does also depend, generally speaking, on I don't wanna say something wrong, but it does depend on the level of the noise in the data already, right? So it is if the data quality is good enough, and probably we did have high quality data in chapter 3. This bias can affect, but wasn't to the point that we were driven, towards extracting wrong information. And the other thing is that there is, in the EBA, while there is a preference for the simplest model, the other two models are still represented a little bit. Pointing towards the fact that specifically if we look at the gradient, that was exactly the reason why I made that map, to kind of have some sort of visual insights in how this variance was divided across the cortex. So while the correct way is to adjust for these things, and there is some literature that came after this study, showed I will take into account this in future analysis, definitely.
Okay. Thanks a lot. Maybe I would like to follow then on the same issue, let's say, but in chapter 4. Here, actually, you have something else, which is the categorical model, which is only one feature. So maybe turning it around, do you trust that most of the voxels where the categorical model is tagged as the winner is actually where this model is the one that explains the most, or is just that the model that doesn't do it as wrong as the others, given the overfitting?
Yes, I would. As I said, this is things in chapter 4 is still under revision, so I do want to kind of like recompute the coefficient of determination to kind of see whether this is still true. But if I, in the case like the categorical model was especially, to kind of take into account the fact that these like very much different things driven by like semantic label can still be represented. But I do understand that, and the reason why we did the analysis that we were trusting the ability of the bounded reach to properly regularize the feature space in a way that this could, you know, kind of be taken into account. But we do need to kind of look into it to follow up on this by recomputing the coefficient of determination.
Giuseppe Marrazzo, the time available for defending your thesis has passed. The jury committee will now withdraw to discuss the quality of your presentation and your thesis, of your defense and your presentation and your thesis, and I request that you and your company remain here until we return from our deliberations.
All rise, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. Southside, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Oh on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
seated.
Giuseppe Marrazzo, the degree committee here present has assessed the quality of your thesis in your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor De Gelder is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right, please.
First question. Do you promise to work in accordance with the principles of scientific integrity at all times?
Yes, I promise.
To be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Then we can continue. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Giuseppe Marrazzo, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and other members of the committee, and affixed with the official seal of the university.
Please be seated.
Well, first of all, congratulations for a thesis completed and successfully defended. We all agreed that not only that the thesis was completed, that was difficult to disagree about, but we agreed that the defense was successful. Your thesis has a challenging and meaningful topic. Meaningful because we work with real life stimuli, with real stimuli. What could be important, more important for you, for social interaction, for human communication, than the signals provided by the human body? It's also a challenging topic, partly because you made the challenge in yourself. You decided to go for challenging methods, and you have our full approval and admiration for that. You arrived in the lab about 5.5 years ago, I think it's actually 6 years ago, and you arrived with an open mind, very eager to learn. You persevered through some challenging passages, some challenging moments, as often they are in a career for a PhD, in a budding career for a PhD student. I remember, for example, the painful revisions we had to do for paper number 1, chapter 2, how at one point we had discussions and I had to convince them just let it go, this reviewer will come back again and again. I think in the end we got like an extra reviewer or something impossible. The reviewer mixed up, the editor mixed up the reviews, etc. It was a bit complicated. Other things went a bit more smooth, and the longer we go, the smoother it seems to be going. So that sounds extremely good, and I'm particularly happy about that. Part of this 6 years period was actually COVID, which we tend to forget now. I can't remember we had any particular difficulty communicating, but that may be my bias, because you are a good communicator. When I want to, I can be a very good communicator also, so that worked out very nicely. Actually, you made the most of the COVID period, because I think you were the first one, and if not the only one of the lab, to get new office furniture out of the COVID reglementation. My application for a new desk, for a new office chair is still on my desk, so I should take your example there. Good. You have been very supportive in our group and also in the ERC group, as Ruffin referred to, in the ERC group at large, where we had regular meetings for the last 4, if not 5 years, and that was very good. I particularly also appreciate that you stand up for your colleagues, even if times were a little bit difficult and some circumstances were not entirely expected. You made sure that, as we say, the train arrived on time and that everybody got their due and got things done that were in the interest of our common work. You even managed to drive one night, the whole night, to Tübingen, because for some obscure reason, I must still say, you absolutely had to play a concert at night, and then you took the train to drive the whole night and be on time, if not early, for our meeting in Tübingen. So that's all very good and very, very admirable. Of course, 6 years is a long time. I'm glad we could afford 6 years. That may not always be the case, or the grant situation evolves goes south, as we say, but so far we could manage that. I'm particularly pleased to grant you this PhD because, as you know, luck comes and goes, love comes and goes, but a PhD is forever. Nobody can take that away for you. I have confidence that you will face future challenges. You have the right approach. Things need to move, things need to get done. And I very much have appreciated that all the years we worked together, and we will still work together some more. Congratulations to your family. I'm sure that some more exposure to your family will improve certain skills you have, some of which I appreciate, like your cooking skills. Although I must say there is room for some more learning in the pizza domain. Yeah. Good.
Okay, I just do a very beginning in English just to explain to everybody that I'm going to do these things in 2 languages, because some of you may know that I am also Italian. Caro dottor Marrazzo, congratulazioni, finalmente ce l'abbiamo fatta. Ho pensato di fare una parte di questo discorso in italiano soprattutto per poter fare congratulazioni alla tua famiglia e ai tuoi cari che sono venuti fino a qua a Maastricht. Siete stati pure fortunati, più o meno il tempo ce la siamo cavata, va? Sì, non ci lamentiamo. Infatti è un momento da celebrare, ce lo dobbiamo ricordare perché secondo me è importante, e personalmente vorrei ringraziarvi, a voi, non a te. Da figlio all'estero io so che cosa vuol dire stare lontano dai propri figli che sono lontani. Mia madre mi chiama tutti i giorni, più di una volta al giorno. Per noi qui a Maastricht avere Peppe è stato veramente una fortuna. Per me personalmente è stato voler dire trovare un amico dopo tanto tempo, e Peppe è stato veramente un amico per molte persone e anche per me. Per questo vi ringrazio e faccio anche a voi le mie congratulazioni. Adesso però devo passare all'inglese perché sennò non ci capisce nessuno e poi stiamo un po' a posto. Dear Dr. Marrazzo, as I said in Italian, my congratulations extend to your family and loved ones. It has been already a long journey, and I'm sure you have had unwavering support from them along this way. A few years back, you came to Maastricht. I think the connection was Professor Elia Formisano that has had a lot of Napolitan students coming over, and many of them coming from the engineering faculty. I don't know why you ended up working with me. And during that time, we looked already at some of these issues about receptive field mapping and so on and so forth. And from the very beginning, I had this impression of having met a very kind person, hardworking person, and a very well-prepared student. Every time I asked a question, Pepe came back with a lot of pages of equations, solving everything and showing me that, in fact, he understood not only the code, but the math that was behind. I was not the one to impress for that, I think. At the end of that period, I don't really remember how it went, but I only remember that because we were talking about future positions as PhD candidates, and Bea had just received the ERC Synergy grant, I tried to make this marriage happen. Given your engineering background, I really thought that this could be a good thing. You then moved here, and I think that the defining moment of the whole move was the fact that you met Leo. Believe me, at the beginning, I was a little bit worried. A guy from Venice and one from Naples take an apartment together in Maastricht. Seems like the beginning of a good joke. I'm not going to end it, but you know, it's how they normally are in Italy. I was wrong, of course. It has become very clear to us all that you have been more than friends, and it is really beautiful to see how much you have supported each other through the years. So it happened that I had the pleasure to follow you a little bit during this career. You moved to using 7 Tesla, developing project using linearized encoding, always under the very attentive and careful supervision of Bea, always with some good pragmatism about the necessity to take choices and actually move forward, which I appreciate a lot, as you know. We had quite a lot of chats, many of them, a lot of discussions, I would say. A lot of them based on topics of methods, of which methods to use. It always involved Giancarlo and Agustin that were always, from you mentioned as the people that you had to check everything with. And I have no doubt that along the way, you've had a lot of support from your office mate, Marta, and of course, Leo, right? Because he was like there all the time. What is impressive is that every challenge you've met with a smile. I think this really, really is the defining characteristic of you. You're always happy, or at least positive, and this is really good. The future holds many more nice things to come. I hope we will continue to share some of them, and I will be happy to come to you for a change when I need some help. Now, let's be done with the professional stuff. I think the whole of Maastricht knows where to go to eat pizza. I disagree with Bea on this. Casatiello, i cantuccini di tuo papà, che sono una cosa spettacolare, voglio ringraziare. A me arrivano tutti gli anni e sono veramente incredibili. When I had to find the recipe for pizza, I struggled for a while, you know, because I was convinced that I needed to use sourdough, and then Pepe told me, forget it, just use this one. Since then, it works all the time. Michelle and Nicolò in particular really love it, and if the reason why they are so happy about it, it's all thanks to you. I shouldn't talk too much about food, otherwise I get hungry. There are a lot of things that I would like to remember. I think the one that I would like to bring up is the fact that at some point, I remember on your black whiteboard appeared the table with some kind of schedule for drinking, eating, drinking water, right? How much not drinking water, how much exercise, these kind of things. You were doing a competition with Marta, and I don't remember who else, so only Marta. And I think I've never met an Italian person from the South being this disciplined. I thought that Marta would win hands down, but in fact, you were close. I don't know whether you know who won at the end. I didn't pay attention to that.
I don't know.
Yeah. So another thing I would like to remember is that you may not know, but last year I organized a thing and I needed help from somebody. I told Pepe something very simple. Can you help me? My neighbors want to do a party. They want to make pizza. I don't want to do it alone because if I fail, it's going to be a big problem. Can you come and help me? It was the end of August. I thought, maybe it's going to rain. Let's see what happens. It ended up being a 30-degree plus day. We were sweating in front of this oven at 600 degrees. And Pepe did not flinch at all, just helped all along the way. The kids ended up, also Oliver, in the pool of the neighbors afterwards. I think we wanted to go in the pool as well, right?
Yeah,
I was really jealous.
Want to, yeah.
It has been a great time. Good friends are hard to come by, and one needs really to hold them onto them tight. I personally hope we will have many more great moments to share. Congratulations again. And now let's go to eat and drink because I am hungry. So.
Dr. Marrazzo, also on behalf of the faculty and the university, I would like to congratulate you with the degree you have earned. I also want to thank the family, the friends, the partner who are all present here, because of course a PhD is sometimes lonely work, but at the same time a PhD you never get all alone. So thanks to all who have supported you in this endeavor. Before I close this session, a few practical points. There will be a reception which will partly alleviate your hunger, Professor De Martino. But before we go there will be some pictures. So everybody will go except for you and your paronyms and those present here for pictures here and on the stairs. There will be enough people who actually know where the reception is, so there will be a stream of people. So if you don't know where it is, you just follow the stream. And with this, I close this academic session.